pull some material together that we've been discussing over the course of the semester and how it relates to insulin signaling. So primarily, I want you to think about insulin in the process of making a protein and modifying that protein and how to secrete that protein. But we're also going to think about hormone signaling and the signals that allow the pancreatic beta cells to secrete insulin. So a couple of times in this video, I'll recommend that you pause the video to give yourself some time to answer some questions. So insulin signals through an endocrine signaling mechanism. So that means you have a cell that generates the hormone, secretes that hormone, and it's transported in the bloodstream to a target cell that has a receptor for that hormone, and where that hormone accomplishes its job. So this is in contrast to the other types of signaling that are illustrated here that we talked about earlier in the semester. So this is a histology image of the pancreas, and it has a few cell types illustrated here. And I just want to briefly talk about the different cell types of the pancreas and their role. So the pancreas is primarily made up of exocrine tissue. So exocrine tissue generates and secretes materials that are used in digestion. And this is actually about 90% of the pancreatic tissue. So the pancreas secretes bicarbonate to help neutralize acids during digestion. It can secrete enzymes that are important to process things. And then the other remaining portion of the tissue is endocrine tissue. So the endocrine tissue generates and secretes hormones. Remember, these are not steroid hormones, just hormones that have other types of receptors. So you may be familiar with beta cells. They're probably one of the more famous cell types in the pancreas. They generate insulin. The other cell types that you may not be quite as familiar with include alpha cells, so they secrete glucagon, and glucagon promotes glucose release. So insulin is secreted in response to a meal, and it signals to cells to take up glucose, and I'll go through that process in a second. And alpha cells you can kind of think of as the reverse, so they signal to cells to take up and release glucose. Other cell types in the pancreas include F cells, and these make a pancreatic polypeptide. This regulates other secretions from the pancreas, and delta cells, which secrete a hormone called somatostatin. Glucagon and somatostatin both signal through G protein coupled receptors, so you should have an idea about what that means when those molecules bind to their G coupled protein receptor. And insulin signals through a receptor tyrosine kinase, which we'll come back to at the end. So the islet of Langerhans is a structure within the pancreas that contains these endocrine secreting cells. So this is an immunofluorescence using antibodies and fluorescent molecules to visualize where cells are that secrete specific hormones. All of the blue dots are cell nuclei in this islet. And you'll notice that glucagon in red and insulin in green are formed and secreted by different cells. So remember, beta cells secrete insulin and alpha cells secrete glucagon. So you'll notice that most of the cells in the islet are actually secreting insulin. So let's talk about how insulin is made. So it's a secreted protein, therefore it has to be trafficked to the ER, through the Golgi, to be secreted from transport vesicles. So this is a schematic of insulin with a signal peptide. So this is the ER signal sequence, a beta chain, a C chain, and an A chain. And you'll notice a couple other amino acids indicated here. So this schematic is showing you pro insulin. And these prefixes in this word suggest that insulin needs to be further processed after it's made to make insulin. So pro insulin has to be processed in a couple different ways. So you should pause throughout this explanation to think about where these modifications happen. So one modification that occurs here to generate proinsulin is that the signal peptide is cleaved. So where is the ER signal sequence cleaved for a protein that is destined for secretion? The other modification that occurs to pre-proinsulin to form proinsulin is 2 disulfide bridges are formed between amino acids 19 and 67 and amino acids 19 and 80. Where do disulfide bridges form in the processing of a protein? All right, so to form insulin, the mature insulin molecule that is secreted, you then need to cleave off this C chain to form insulin. And so there are enzymes in the Golgi that recognize these basic amino acids next to each other, arginine and arginine, lysine and arginine, and that enzyme recognizes those and cleaves off the C chain from proinsulin to form mature insulin, and this occurs in the Golgi. So the C protein has other functions in the organism. It can signal through a calcium-dependent pathway and has been implicated in diabetes neuropathy. So that means symptoms in diabetics that are related to the nervous system. So diabetic neuropathy. So some patients who also need to take insulin may or may not make a little bit of insulin. But the insulin that a patient takes does not have to go through this processing. It's already available for cells to detect in the bloodstream. So the difference between the insulin a patient takes and the insulin a patient might make, one thing is this C chain. So doctors can detect the amount of the C chain in the bloodstream as a proxy for determining how much insulin the patient is making on their own. So you can look at how much C chain is in the patient and compare that to how much total insulin is in the patient. Because for each insulin molecule a patient makes on their own in their beta cell, they'll make one C chain that's cleaved off and also secreted. All right. So this is an image of a secretory vesicle containing insulin here, and one secretory vesicle that has already fused with the membrane. Remember, insulin is secreted in response to a meal and sugar in your bloodstream. So what is the process by which a cell can do so, can detect the sugar and know to secrete insulin? So I'm going to walk through these steps even though they're all written here, and here's the image, that electron micrograph, again showing you that secretory vesicle and the fused secretory vesicle. So the resting state of a beta cell is that you have voltage-gated calcium channels that are closed and ATP-sensitive potassium channels that are open. So based on what you know about ion balance at the cell membrane, if ATP-sensitive potassium channels are open, which way do you expect potassium to flow? And remember, potassium flow and concentrations are what determine the resting membrane potential of a cell. So once you have a meal, glucose is transported into beta cells through facilitated diffusion of glucose transporter. You know from lab that cells take up glucose and metabolize that glucose to make ATP that the cell can then use. So when a beta cell takes up glucose, starts making a bunch of ATP, what do you think the next steps are based on this resting state of the beta cells, where you have ATP-sensitive potassium channels and voltage-gated calcium channels? Should pause the video here and sketch out a flow chart of what would happen. All right, so now the cell knows to fuse those secretory vesicles and releases insulin. At the target cell, there is a receptor tyrosine kinase. This is the receptor. Remember, there's 2 subunits, and in red here is the insulin. And once the cell binds to the insulin, it knows to open glucose carriers and take up glucose in the cell, only when excess of glucose is available. And this is important because the brain regularly takes up glucose, and so this allows the body to make sure there's always enough glucose for the brain to take up, and then insulin-dependent tissues only take up glucose when there's extra glucose in the body. So this is really important for glucose metabolism and regulation in the body.